Welcome to Let's Talk Micro. Hello, everyone. Welcome to another episode of Let's Talk Micro. As always, I hope you had a great week. And you can always find Let's Talk Micro on Apple Podcasts, Spotify, Google Podcasts, Amazon Music, Pandora, Goodpods. Whatever you listen to podcasts, you can find Let's Talk Micro. As for our social media, I am on Instagram, TikTok, and YouTube as Let's Talk Micro, on X, which used to be Twitter, as Let's Talk Micro 1, on LinkedIn as Luis Plaza, and I also have an email address, which is letstalkmicro@outlook.com. So please subscribe to the podcast, download episodes, leave a review if the app allows you to do so. And if you have any feedback, any suggestions, like any topic you think you might make a good guest, you can always reach out via social media, or you can email me at letstalkmicro@outlook.com. If you haven't listened to the last episode, the latest one, please go ahead and do so. Season 3 kickstarted with a great episode on the AMR subseries, which was episode 2, and this one was about beta-lactam antibiotics. So, as I said before, in this series, Dr. Andrea Princey is the co-host, and we had a guest, Dr. Brian Rowe. So we talked about the beta-lactam antibiotics, we talked about the mechanism of action, which in the case of the beta-lactams, they target the cell wall, and it's not the only class of antibiotics that does this, but it uses a different pathway. So we talked about that. We talked a little bit about the differences between the cell wall of gram-negative organisms versus gram-positive. We went over the beta-lactams. We talked about penicillin, carbapenems, cephalosporins, and degenerations. We talked about the CLSI, and we talked about the Appendix B and intrinsic resistance, and how is that helpful when we are, how does that relate to beta-lactams, and why do we need to know this information? So overall, it was a great episode. It was very informative, and down the line, we'll continue publishing more episodes about on the AMR subseries. So I hope you enjoyed it, and if you haven't listened to it, please go ahead and do so. So in today's episode, we talked about Francisella, and thankfully we don't see it a lot in the lab, but we are trained to recognize it, right? And has the potential of being used as a bioterror agent. So two guests come to the podcast, Dr. Ryan Relich and Dr. Kenneth Gavina from the Indiana University School of Medicine, and they joined the podcast to talk about a case from blood cultures where Francisella was recovered. So we break it down. We talked about biochemicals, morphology, what should we do when we suspect it, and we also talk about the importance of being able to recognize these organisms and follow proper protocols, to always have a plan in place. Since we rarely see it, sometimes, we go into this type of situations where we recover an organism from a culture and then we are unable to identify it, it's growing poorly, instruments are not given a proper ID, and then we start consulting with people, right? And then we maybe involve a senior tech, a supervisor, maybe another tech, and all of a sudden you have 4 or 5 people that are involved in this, and then you end up getting one of these organisms, and then everyone has to go into some sort of prophylaxis treatment. So we definitely need to keep these organisms in mind when we are working on cultures. We make sure that we follow the proper protocol and we stay safe. So it's very important to review the procedures of the lab, and I mentioned also the resources, right? Like the ASM handbook, which tells you when to suspect organisms like this. So overall, it was a great episode with Dr. Brian Relich and Dr. Gavina, and both of them, they're going to be coming back on the podcast with other topics. So definitely a great episode. I hope you enjoy it. So let's go ahead and listen to it. So on today's episode, we are here to discuss an article and a bug that is very significant, that we don't see that often in the lab, but we should always be prepared for it. So this article is titled "The Briefcase: Suspicious Gram-Negative Coccobacilli, Francisella tularensis, subspecies novicida, isolated from an immunocompromised patient." This was published in the Journal of Clinical Microbiology in June of this year, and this is a journal from the American Society for Microbiology. So with me today, I have two guests, Dr. Kenneth Gavina and Dr. Ryan Relich. Doctors, welcome to Let's Talk Micro.
Thank you.
Hi, Louise. Thanks for having us.
Definitely my pleasure. So for the audience, can we go ahead? Let's start with a quick introduction, like what do you do and anything else that you want to tell the audience?
Absolutely. Ken, why don't you go first?
Thank you. Sure. So my name is Ken Gavina. I am the Director of Clinical Microbiology and Serology here at Eskenazi Health in Indianapolis. I'm also an assistant professor of clinical pathology and laboratory medicine with the IU School of Medicine. In terms of a little about me, I am a recent CPEP graduate and new to my role. And I had a wonderful mentor with our co-guest today throughout fellowship. So I'm very excited to be part of this episode with a good friend and mentor of mine.
All right. Well, thank you, Dr. Gavina. And I am Dr. Ryan Relich. I am the medical director of the Division of Clinical Microbiology at Indiana University Health, as well as the medical director of the Special Pathogens Unit Laboratory, and interim medical director for the Division of Molecular Pathology. And in the not so distant future, I will also serve as medical director of a new donor screening laboratory for stem cell donors that we're opening here at the IU Health Pathology Laboratory in Indianapolis. In addition to that, I am an associate professor of clinical pathology and laboratory medicine and an adjunct associate professor of microbiology and immunology at the IU School of Medicine. And I have a research laboratory. It's basic research and translational research. I study emerging viruses, so high containment viruses that require BSL-3, things like MERS coronavirus, SARS-CoV-2, and a variety of arboviruses.
Well, once again, thank you for being here. Both of you, I kind of just through social media, I've seen you before. I had the chance to meet you in person at ASM. I think Dr. Rich, years ago, I saw you in a conference. I didn't speak to you. I just saw you speak, but we had a chance to chat briefly at the ASM. Okay, so let's go ahead and start with a quick overview of the article.
Yeah, maybe I can start us off on this one. So this was a case that we saw here at one of the hospitals within our IU health system. It was a 63-year-old immunocompromised male who presented to the hospital with acute pancreatitis, had a very interesting case history. So the patient had a past diagnosis of blastomycosis, which is a dimorphic mold that we see here quite frequently in the state of Indiana. Had tobacco and alcohol use, but also had what he described as a frequent outdoors or woods living activity. So this patient in particular would live outdoors, kind of off the wilderness in his backyard, adjunct to his house. So he was really kind of the clinical microbiologist or infectious diseases dream of a case study with all the possible differentials that you could have. So he was admitted for acute pancreatitis, and subsequently his blood cultures, when they came to the laboratory, were isolated for a Francisella organism, which kind of led to us publishing this paper as a case study, mostly on the case of laboratory safety and why it's important to be able to recognize these organisms, but to use as an educational resource. And it's also just quite a fascinating story itself.
Yes. So as I was reading the article, so as the point of view of an medical lab scientist, working with plates, day in and day out, a lot of times, we learn about these organisms in school. We don't see them as often in many areas, and then we do, we get like a proficiency service every year in the lab to train for it, to recognize them. But sometimes, people don't really think of them right away when you're getting your workflow is not growing. You go through like reviewing a smear, maybe resetting it, maybe add an additional plate if it's needed. So when did the laboratory start thinking of the possibility of a biological threat agent?
Yeah, so Dr. Gavina and I were consulted on this case whenever the gram stain was reviewed. There were small gram-negative coccoid organisms present within the blood culture gram stain. And at that point in time, there wasn't a whole lot of consideration given to the possibility of Francisella. The organisms were actually a bit large, we thought, for Francisella tularensis. And so the subculture plates were read at the bench by the technologist, who then later alerted us to the fact that all of our conventional diagnostic methodologies that we use for microbial isolate identification failed to yield an identification. And so at that point in time, we got concerned that, oh boy, maybe this is Francisella. It just looks a little bit different than what you would expect from the textbook photographs. I have some preserved slides of Francisella tularensis and compared the two, and they look nothing alike. And so subsequently, out of an abundance of caution, we decided to handle these cultures as if they were a select agent. And eventually, the cultures, after going through as much rule out testing that we perform in house, they were submitted to the Indiana Department of Health laboratories for additional testing. They serve as our select agent reference laboratory as part of the laboratory response network. And so they did the additional physicochemical testing as well as genetic testing to render a preliminary identification of Francisella tularensis.
Okay, yes, and it can be, sometimes, yeah, like with the service that we get, we kind of like prepare and then we're going over, we get case studies with the samples and we're kind of just ruling them out. But yeah, I guess in the mix of cultures, yeah, after initially it can be a little difficult to start thinking about Francisella, but as you were saying, yeah. So let's go ahead and for the audience, if you're just tuning in and this is your first episode, I did an episode with Yersinia that you can go back. And I actually, if you have questions about what the laboratory response network is, you can definitely go ahead and check those episodes out and I go over them. So let's go ahead so the blood culture ended up having Francisella. So let's go ahead and just break it down, biochemicals, reservoirs. How do we acquire it? How do we ID it? So let's go ahead and do that.
Sure. So I think to Dr. Relich's point, typically what will happen is at the time the blood culture bottles flagged positive off the instrument. So we pulled them off at our institute. We were using the Bactec FXs, and we performed a direct gram stain from the bottle. So as was mentioned previously, these were some smaller gram-negative coccobacilli, although to his point, they did look a little bit big of what typical Francisella species would normally look like. From there, our reflex test at the time was the Veregene blood culture gram-negative panel. So we threw the isolate onto the Veregene instrument, which failed to yield an identification. From there, routinely, we would then move to our culture plates. So at the time, we use a blood agar plate, a chocolate plate, as well as a McConkey agar and a CNA. We then saw growth, I believe it was after 48 hours of incubation on the plates on both the chocolate and the blood. And to kind of further muddle the mixture, so to speak, these colonies were also pretty atypical of what normal Francisella would look like. We would typically expect these colonies to be a little bit slower growing. We would expect to see them on the chocolate agar and maybe scant growth or no growth on the blood agar, but these were noticeably larger than what we would typically see from a Francisella isolate. So from here, we would move on to some of our more traditional biochemical methods. And as you mentioned, there are some fantastic resources on APHL, which is the Association for Public Health Laboratories. They provide a sentinel card resources for different select agents and biothreats, and Francisella is certainly chief among them. But some of the biochemicals that we're particularly interested in looking at when we're looking at these gram-negative coccobacilli would be the oxidase test. And we would expect Francisella to be negative in this case. Another biochemical test that we can perform that's fairly rapid and easy is a catalase test. In this case, we would expect it to be negative or weakly positive. And it was weakly positive in the case of our case here. And then a urease test, which we would also expect to be negative. Additional tests that might be helpful for the identification of Francisella would be a beta-lactamase test, which could be positive, and then a satellite test, which is helpful for differentiating Francisella organisms from Haemophilus influenza. So a satellite test, as I'm sure you've talked about before in previous episodes, and many of our listeners are aware of, are looking at our X and V factors, which is our NAD and our Heman factors, and the requirements for growth. So with a satellite test with Francisella, we would expect to see growth independent of these discs, whereas Haemophilus influenza, we would expect to see the growth kind of isolated to the X and V factors that are required for growth.
Yes, and thank you for that. I have definitely talked about it, and it's always nice to provide that visual when students see the colonies around. And it's even greater when you see it in an actual culture that all of a sudden, you see the little dots around the Staph aureus, and then let me take a look at that chocolate, and boom, there it is, all nicely grown for Haemophilus. So as far as, as I was reading the article also with identifying it, sometimes some platforms, it can be a little misleading. So in this case, it was ultimately identified by, was it by PCR? And then, so how do we normally identify it? This is the way to actually identify it. I mean, at this point in time, yes, when we suspect Francisella, we use our laboratory response network and we send it, which ends up being, your health department from the state. And so is this how it is identified?
Yeah, at our institution, suspected isolates of select agents get sent to the Indiana Department of Health laboratories, who are responsible for performing additional characterization. They did end up sending this isolate on to their reference laboratory, the top tier lab, CDC, who provided the definitive identification of Francisella tularensis. However, one thing that was sort of a surprise for us was the fact that it was the subspecies Navicida. So there are 4 subspecies of Francisella tularensis. There's Francisella tularensis subspecies Holarctica, Mediasiatica, Novicida, and subspecies tularensis. And of those 4, Navicida is the one that you kind of hope that you get in the laboratory because it's not a select agent, and it is a risk group 2 pathogen, which is then able to be handled using ordinary standard laboratory precautions at biosafety level 2. Whereas the others, you ratchet up the biosafety work practices and physical containment practices, and you also try to ensure, that for biosecurity reasons, that all of the culture specimens and other materials associated with the culture are well controlled and locked away and ultimately destroyed, and that destruction being documented and proof thereof being submitted to the DSAT, the Division of Select Agents and Toxins at the Centers for Disease Control, within 7 days of destruction. And so, fortunately, the whole genome sequencing that CDC did on this isolate indicated that what we were working with was sort of the diet version of Francisella tularensis, which didn't pose much of a laboratory hazard to our technical personnel. And so from there, once we got that information back, we sort of stopped all of our workup that we would do in house for exposures to potential select agents and other, risk group 3 type of agents or others that present a high likelihood of occupationally acquired infections, because there's not really any indication whatsoever for doing any type of post-exposure testing or post-exposure prophylaxis or anything of that nature. However, had this been, subspecies tularensis, we would have filled out all of the appropriate paperwork and did the post-exposure prophylaxis. And we work with whatever we have instances that require that type of attention. We work with our occupational health physicians and other care providers to sort of drive that post-exposure workup.
Okay, yes. And as we are talking about it, I like, by the way, that you use that term, the diet version of it. I'll remember that. So as far as, you mentioned that the patient had an outdoor living style. So maybe for the audience, can you talk a little bit about, and this is mentioned in the article if anyone wants to read it, which I will put on the show notes. So as far as, reservoirs, where can we find Francisella?
Yeah, that's one of the beautiful and scary things about Francisella is that it is a zoonotic pathogen with a wide host range. And in the United States alone, there's been reported cases in almost every state with the exception of Hawaii and Alaska, I believe. It's generally transmitted by arthropod vectors, so ticks, specifically like the dermacentor ticks, but you can also see it in amblyoma ticks. They're also aquatic vectors too, with like the deer fly and mosquitoes that you can see through voles. Um, it's commonly taught or referred to as rabbit fever. So rabbits, and then I think one of the case stories that we'll often hear in training is the lawnmower incident, where a lawnmower kind of runs over a nest of rabbits and almost aerosolizes the Francisella into the air, which again is one of the major reasons why it's so infectious. So to kind of get to that point, the infectious dose with Francisella, for depending on the subspecies that you're dealing with, can be as low as 10 colony forming units or 10 bacteria, so which is quite low compared to some of the other infectious doses of other infectious organisms. But in addition to zoonotic exposures, so again, you can have contact with infected animals or carcasses. You can have certainly the arthropod vectors leading to bites or exposures, but you can also have ingestion through contaminated food or water. The inhalation of infectious aerosols, whether it be within a laboratory or dealing with these animals, is certainly a high risk exposure. You can have professional exposures, or like work class exposures, such as being like a veterinarian working with these animals, or a farmer, or even ourselves as laboratorians having to work up with these specimens without appropriate biosafety and PPE and precautions.
Okay, and excuse me. So as you mentioned the biochemicals and how it should look, just to kind of just tie it all up together for the audience. So when you're working with these, right? Sometimes, a lot of places, the initial stain is done by one area, especially very large facilities. Someone else is working the plates. So when should we start thinking, maybe this is Francisella?
Really, at the time that the gram stain smear from a blood culture, another perhaps a clinical specimen, are interpreted, oftentimes discerning these bacteria from proteinaceous debris and quote background material can be difficult. However, in the case of blood cultures, it's typically, I don't want to use the word easy, but these organisms are noticeable. And if you have a good quality microscope, you can even distinguish individual cells from one another. And so whenever you see these tiny gram-negative coccobacilli, definitely that should serve as a warning to you that it could be Francisella tularensis, or it could be Brucella, one of the brucellosis-associated Brucella species. Let's not talk about the Ochrobacterium brucella nomenclature thing, but with Francisella tularensis, you should definitely have heightened suspicion whenever you see that. And subsequently, there should be system in your laboratory in place for communication of that information to the other laboratorians, as well as the supervisor, medical director. And it's really up to those folks to work together with the laboratory staff to ensure that the appropriate precautions are taken and that the appropriate rule-out testing is done. And the rule-out testing, if your laboratory has a biosafety level 3 laboratory, that those cultures can be taken into and manipulated within a class 2 type B biosafety cabinet while wearing respiratory protection and sort of the other BSL-3 garb, then that should be done. At the absolute minimum, in laboratories that don't have any type of containment space, work should be done always in a biological safety cabinet while wearing the appropriate personal protective equipment. And in addition to that, all of the wastes that are generated as a consequence of culture manipulation should be sterilized by autoclaving or soaking in a disinfectant for the appropriate time. And now, to put your safety hat on, also one of the things that you should do prior to doing any of this work really is a safety risk assessment. Determine sort of where the weaknesses are, determine what type of precautions are necessary for containment and are also necessary for abatement of any issues that may be encountered along the way. You should really have in your standard operating procedures within your facilities protocols that address what happens if there's a breach of containment. What do you do in the event that there are exposures? Because things like that happen. I mean, busy medical laboratory scientists working in microbiology, maybe working up 400 cultures or more in a large laboratory. And you need to get your work done. The clinicians are relying upon us for the expeditious reporting of results whenever they're available. And so the technologists then are charged with the task of doing a whole lot in a short period of time. So it's easy to just sort of disregard these things. And perhaps, the first thing on their mind isn't Francisella, Brucella, or another select agent, but just some run-of-the-mill microorganism that they see on a routine basis. And so it's really important, too, to have periodic training sessions and refreshers or, quote, in-houses in order to ensure that everybody's on the same page, and have abundant resources available to the technical staff as well.
Yes, I definitely agree, and it can be. We definitely, yeah. Sometimes, there's a lot of volume, and yeah, it can definitely. So the importance of training, PPE, a plan in place, yeah, because a lot of people sometimes, they're not thinking about this, but definitely, right? There should be, especially like you said with the gram stain, and if you start seeing that maybe the growth is not, it's not growing as healthy. It's like a slow grower. Maybe you should start considering that because you have sometimes coccus bacilli, your Haemophilus, it will definitely grow very strong when you sit down and look at the cultures. The same with the Pastorella, very healthy on your blood, on your chocolate. So those are also things to keep in mind as you're working this. And the communication, yeah, you definitely you said it. Sometimes even we had cases with Brucella where it's actually it's been suspected, and we function as a reference lab for other labs, and no one says anything. And then you got a tech that's opening. I have this, it's growing poorly, it's not identifying, and then the next step is, well, let me ask maybe the senior tech. So that's two people, and then maybe ask the supervisor. That's three. So you're stuck. Let's ask our director. So all of a sudden, it comes out as Brussels, and then you have all these people involved that they were exposed. So definitely, like you said, training, communication, and PPE. A lot of people, most if you're a micro student and maybe or a medical lab science students, a lot of these plates, your routine plates, are worked out in the open. So if you start thinking about this, definitely follow the proper protocols. So these are all the, I think this case, it brings all these important facts to light. Is there anything else that you think about, that's very important that comes from this case?
I think you guys really both hit the nail on the head in terms of communication, not only between personnel in the laboratories or interlaboratories in the case that you mentioned, Louise, between like a reference laboratory where they might suspect it, but also for our clinical colleagues as well, to kind of give the laboratory a heads up if the clinical picture makes sense. If you have a patient who maybe is a returning traveler who's been eating unpasteurized dairy for the last 2 weeks and suddenly develops this undulating fever, maybe give us a heads up, because that's something that can definitely put it on our radar as well. And to your point, Louise, as far as like it is very common practice in the microbiology laboratory to get a second, third, fourth, fifth opinion when you're unsure of something. It's the great thing about keeping an open dialogue and communicating. Unfortunately, it's kind of a double-edged sword when you're dealing with something that's highly contagious and it's not being handled with the proper precautions. So I think one of the key aspects that I think this case highlights is that alone is being able to recognize your rule out algorithms and proper biosafety procedures. An example of this is the reliance that most modern day laboratories have on MALDI. So MALDI-TOF, in a sense that we become so reliant with just throwing the organism and spotting it onto our MALDI plate and then throwing it on. However, when we come across a sample that maybe doesn't give us good peaks or maybe a suspicious ID, what do? We tube extract it, we vortex it, all these additional steps which create additional exposure risks to the personnel in the lab, and typically are also done on an open benchtop. They're not going to be often done in a biosafety hood. And if you're in a situation where your laboratory is set up as like open bays, not necessarily these isolated rooms, you're now creating these aerosols, which not just impact the lab tech itself who's working that isolate up, but everyone else around that lab tech as well. So I think certainly the highlights for me for this case was definitely recognizing bioselect agent rule out algorithms and kind of keeping an eye and keeping an open mind about the fact that how our reliance on some of these automated technologies and rapid diagnostics such as MALDI-TOF and like even the blood culture identification systems, there's always going to be a room and a place for rapid biochemicals. And I think that's kind of what this case here highlights.
And even too, to that end, morphological observation. I'm aware of some laboratories that no longer do the first stop as a gram stain from a positive blood culture. Instead, they'll just blindly put it on their multiplex molecular panel, which could potentially create biohazardous aerosols or requires sort of unnecessary manipulation of the cultures, which we try to avoid whenever we have circumstances um, dealing with select agent cultures. And so good practice is to always start off with a gram stain. That can help influence the decisions you make about, what types of things may be happening. And, the way I kind of look at this is you're essentially establishing a profile for these organisms, like a dating profile. You make this list of things that you're looking for, and then you start checking off boxes. And once you hit a certain number of boxes, then you go, oh, this could be this or this could be that. And I think chief among the things to do early on is do some of the simple things like growth rate determination. And yes, it's growing luxuriantly on chocolate agar after 72 hours, but the colonies are very tiny on the blood agar plates. And, the cellular morphology and its size and gram stain reaction are consistent with Brucella, Francisella, whatever. So even that type of thing is very, very important. A lot of these newer technologies are absolutely fantastic. They have been solving problems that we've been suffering with for many, many years. However, they are just tools in a toolbox that also include some of the more traditional approaches that our medical laboratory scientists, supervisors, and medical directors must absolutely carry into the future. They must continue to be stewards of that knowledge and judiciously apply it to the laboratory diagnosis of infectious diseases. And the last thing I'll say is that with regard to this case, it just goes to show how important it is to have a good working relationship with public health. All too often, the public health laboratories may be overlooked in cases like this. However, they are the right hand, we're the left hand, or vice versa. You should have a very, very good relationship with your public health laboratories or and reference laboratories and other folks. We're all on the same team. We're working to accomplish a common goal. So, maintaining a good relationship or establishing and then maintaining a good relationship with your public health laboratories will ensure that, things go as smoothly as possible. And just a plug for public health also, they oftentimes will offer free training for packaging and shipping of biohazardous substances, infectious agents, as well as, like select agent 101 type of training, as well as PPE training, work practice training, things of that nature. So those are oftentimes free resources that you and your laboratory personnel can take advantage of in order to, help bolster your own safety practices and work practices in the clinical microbiology laboratory.
While we're giving kudos, I think we should also give a huge shout out to our IU Health infection prevention and control team, because at the time when we had these exposures, when we weren't sure of the subspecies identification, they did a great job consulting with us in terms of identifying the at-risk or exposed individuals, getting them on the pre-exposure prophylaxis like rapidly, and then symptom watching for the next several days. So honestly, it's such an that's what healthcare is it's such an integrated system with so many moving parts, and we're all, and to Dr. Relich's point, we're all trying to do the best that we can to provide the best patient care that we can, but that also involves keeping our staff and our laboratorians healthy as well. So definitely big shout out and props to them.
Indeed, it's definitely very satisfactory when, right, when we all work together as a team, and the result is, it's we all work together, and like you have said, communication. And you definitely touched some points, Dr. Relich, and about the gram stain, and this is something that I have talked in so many episodes. Such a helpful tool, and I will continue repeating it to the audience out there, especially if you're just a medical lab science student, getting ready to graduate and start your first job. It is a very helpful tool. So yes, the MALDI-TOF is great, but your biochemicals, your gram stain, it will help you. And like Dr. Roach said, I like that term, the dating profile. So you're putting everything together, and it is so helpful. And sometimes you can avoid mistakes just by taking those steps. Gram stain, biochemicals, and you'll be surprised, how many times you get a consult, you talk to someone, and you ask, "Have you done a gram stain?" And they're like, "No." Okay, so you said it failed in the MALDI 3 times. So gram staining, let's see what we got and start from there, and then we'll make a determination. So it's just very helpful tool. So please, if you're listening, use them, and they're definitely going to help you, and stop you from making mistakes, which, in this field, mistakes, affect the patient. So, well, is there anything else that either of you want to add?
I don't think so at this time, but I definitely appreciate you having us on to talk about this. And Dr. Gavina, congratulations on your new position and for being an excellent CPEP fellow. Just a shout out to all the CPEP programs throughout the United States. Ours is one of 21 or 22 programs in existence now, and so for folks out there that are interested in pursuing training to become a medical microbiology or public health microbiology lab director, that route through the CPEP program, I think, is a really, really great. And so this time of year, these programs, including ours, is accepting applications. So definitely look up programs. I'm not trying to give a shameless plug specifically for IU's program, but if you happen to come here, you get to work with me and Dr. Gavina, and we get to work on interesting cases such as this typhoidal tularemia case. But with that, I've got nothing else. So thank you very much, Luis, for your time. And you and I will be talking again on Monday about emerging viruses. So I'm really excited about that.
Yes, definitely. Me too. And if the audience, if you're listening, definitely stay tuned, because that's an episode I'm looking forward to recording and sharing with you. So some great information coming your way. So this has been, great. And once again, I appreciate both of you taking the time to come into Let's Talk Micro.
Yes, thank you for having us. Greatly appreciate it.
Thank you for having us.
My pleasure. And that, my dear audience, it's the end of this episode. I hope you enjoy learning about Francisella. As always, I enjoy sharing this information with you. As always, continue bringing that passion to what you do. It's so important. We do such great work. Thank you for the support. Please continue downloading episodes. I appreciate it. I will continue bringing more information to you. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.